performance that you just did, I would like you to double it as we welcome Sean O'Byrne to the stage.
Hello everybody and welcome to Science Land. I'm Sean, I'm from Ireland, and tonight I'm going to tell you a little bit about what I do. So I'm a multidisciplinary scientist. I work in chemistry mainly, but I do a little bit of biology and I dip my toe into engineering. So I work with malaria. I presume most of you might have heard of it. Has anyone took medicine for it? Yeah. Has anyone actually had malaria? Yeah. It's not very nice. So malaria, if you get it mildly, you can get fever, aches, diarrhea. It can get worse if it goes untreated. You can get organ failure, which is not very nice. And in the worst cases, you can get coma and death. Now, half the world's population is at risk of malaria. See on the map here, the red areas, population over 60% is at risk. And in these blue areas, it's between 20 and 60%. So it's quite a significant number. So now, oh, what's come up yet? Some stats about malaria. Last year, it was estimated there was 200 million cases of malaria. And there was somewhere between 500,000 and 1 million deaths. Quite a large number. And it's actually been speculated that since humans first evolved, half of all people who have lived have been killed by this disease, which is quite a significant number. Now, part of the problem with malaria is that it can be hard to diagnose in the areas it's endemic. So if you or I thought we had malaria, we'd simply go to a hospital, we'd give a blood sample, and they'd analyze it under a microscope, and they'd see something like this. So these are all red blood cells. And most of these are healthy cells. You can see the round ones that are opaque and see-through. But then you see these ones that are darker with the purple spots. They're cells that are infected with malaria. And this is how you can tell in a hospital or in certain medical centers if you have malaria or not. So that's all well and good. But in a lot of areas where malaria is endemic, you don't have access to things like this. Even if you had a medical center close to you, there's a high likelihood that they wouldn't have the facilities to do tests like that. And what they do is they diagnose you depending on your symptoms. Now that's fine, but the problem is there's a lot of diseases and sicknesses like malaria that have the same symptoms. So if you go to a doctor and you did get treated, it was all well and good, be fine. But if you didn't have malaria and you got the wrong medicine, or you had some other disease and you got the wrong medicine, you wouldn't get better, it'd affect your community, and in the worst case scenario, you'd actually die from it. So this is something I hope to change. I want to be able to diagnose malaria simply, without the need for a doctor, a hospital, a microscope, just somewhere you can do it out in the middle of nowhere. Now, to explain how I do this, first I need to tell you a little bit about the malaria parasite. So you get malaria when a mosquito infects you looking for a blood meal. In a mosquito's saliva, it has some parasites. And when they get transferred into your blood, and then from your blood it migrates to your liver, and it infects a liver cell. Now inside your liver cell, one of these parasites will reproduce, and it'll reproduce up to about 30 times. And once it gets to about 30, your liver cell will burst, and it will release more parasites. Now they go to your blood, and then they infect your red blood cells. And again, quite similar to the liver cell, once the parasites are inside your red blood cell, that's the point it'll start to reproduce again, up to 30 times. It'll burst out of your red blood cell, and more of these parasites are free then to go and continue the cycle of invasion, and the cycle propagates. Now some of you might have heard of the recurrent fever that comes with malaria every 2 days or so. That's the parasites bursting out of your red blood cells. Now it's not very nice. And a really important thing when you think about it is when the parasite's reproducing, it's going to need a lot of energy. And usually, it's quite hard to get things in and out of your cells. They're quite well kept. It's good usually. It keeps the bad stuff in, the good stuff out. The bad stuff out and the good stuff in. So to compensate for this, because the parasite needs a lot of energy, it installs a transporter in the cell surface. And this transporter acts as a highway for the cell's metabolism. It hoovers up a lot of food that's around it, and it gets rid of all the waste that the parasite produces. So what we've discovered is a way of getting things in and out of the parasite using this transporter. It's almost like, well, I call it a Trojan horse, because the parasite thinks it really wants it and really needs it, so it hoovers literally everything around it and just take it all in. And the best thing about it is we can attach different things to it. Now it can be chemicals, there can be drugs or different chemicals. We can attach DNA and we can attach proteins, loads of things. So that's really good. So then once we discovered this, the kind of Trojan horse, it was my job to turn this into a way of diagnosing for malaria. And I think how I've done this is pretty cool. So I went to the lab and I made a fluorescent chemical. Now fluorescent is something that simply absorbs light and emits it again. So do you know some of the kids that have stars on their ceilings, during the day they absorb the light and then at night they glow? That's kind of something similar to what I made. Except what I made glows under fluorescent, or under UV light. And it's called a Bodippi. Now Bodippies are really good. As I said, they glow under UV light, really bright. They're non-toxic. For a load of other reasons, they're really good in biology. Now, I wanted to bring some, but I wasn't allowed because I need a license for chemicals. This is what they look like. You can see, it goes from blue, green, yellow, orange. There's ones that are red and purple as well. So I made one, it's quite similar to this. So then, once I'd made that chemical, what I did was I attached it to my Trojan horse. And I had a fluorescent version of this Trojan horse. So then I went back to a lab, and I went to a biology lab this time, and I brought some of my chemical with me, and I got my hands on some blood that was infected with malaria. So I mixed up the chemical that I'd made, this malaria. I left it for maybe half an hour, or an hour, and then I took it, I washed it, and I put it under a microscope, or put it on a slide, and then I looked at it under a microscope. And this is what I saw. So these are red blood cells again. See, most are healthy. The ones with these dark circles inside of it, they're the ones infected with malaria. So then what I did is I turned off the white light and I shone a UV light onto them. This is what I saw. The cells infected with malaria had taken up my compound, but the healthy cells weren't. Only goes in through that new transporter that the malaria puts on your cells. So at this point I was pretty happy. I was getting a good result. But then I thought of a possible problem. In the 48 hours that malaria is inside your cell and it's reproducing, it goes through 3 stages. It's almost like a child, a teenager, and an adult. Now in the blood I had used mixed stages. So I didn't know whether my chemical was going into the first stage, the second stage, or the third stage. And I got quite worried because, well, I thought this could be a real snag in my system. And I was quite disappointed. So I went back to the lab. With the help of a colleague, I was able to synchronize the parasites. Now all this meant is that the parasites inside of the blood cells were within an hour's age of each other. Now this took a lot of work and it was quite hard to do, but I got it done, and then I tested it stage by stage over the course of 48 hours. It was also quite hard and I didn't get very much sleep, but by 1 AM on a Friday night, I got the result I wanted. I had pictures quite similar to this. So again, it's the white under white light. And this is the first stage. You can see healthy cells. There's one infected cell there. This is the second stage. There's some healthy cells and infected cell. And here, the third stage, quite a few healthy cells and some infected cells. But then, turned off the white light, switched on the UV light, and I saw this. The first, the second, and the third stage. And it worked in all of them. So I was really, really happy at this point. But now you're probably thinking, that's all well and good. You have this, it's great, but you said the whole point was to be able to easily diagnose malaria without the need for a microscope. And all I've done so far is use actually a pretty fancy and expensive microscope. This is where my friends in engineering come in. Now my friends in engineering are experts at making lab-on-a-chip devices. And these are just really small devices that fit in the palm of your hand and are able to do a range of laboratory function. Now they're really cool. They combine nanotechnology, microfluidics, and material science to be able to do this. And they can do a range of things. There's ones out there that are actually quite commonly used to test for HIV. There's others that can measure sodium levels in blood. There's some of them are actually able to centrifuge cells on a little tiny microchip. But there's one in particular that I was interested in, and that is one that contained an optical microspectrometer. Now a spectrometer is something that just simply measures something and creates a spectrum. The one they made measured light, hence the optical part. But what was good about it is they could tune it to measure an exact wavelength of light. And an exact wavelength of light means an exact color. And they're able to tune it to measure this color here, the green at 512 nanometers. So this brings me to where my research is currently. I have this way of testing for malaria that's never been done before. My colleagues in engineering have a way of detecting light. So I want to combine the two of them into one device that makes it easy to test for malaria. And how I envisage this is making something maybe the size of a mobile phone. It'll only take a fraction of the power you need of a mobile phone. And if you think you have malaria, what you'll do is you'll take a pinprick of blood, you'll put it into this, it'll mix with my fluorescent chemical, and then you just wash the blood to make sure you don't get a false positive. You use your microspectrometer to analyze it. If the blood's infected, it'll have taken some of my fluorescent compound in, and this will be seen by and detected by the spectrometer. And then all you need is a simple bit of electronics that converts that detected signal into a visual readout. It could be something as simple as a tick or a cross. Now, to finish up, what I want you to imagine is imagine you lived in a rural area where malaria was endemic. Previously, you had to travel maybe far to go to a medical centre to go see a doctor who possibly might not even be able to tell you for definite whether you had malaria or not. Now, instead, all you have to do is be willing to have your finger cut to take a pinprick of blood, and then wait half an hour, and he can tell you for definite whether you have malaria or not, and you can get the proper treatment you need. Thanks for listening, and I hope you enjoy the rest of your evening.